Hello and welcome to BioCode. Hope you are doing well. The agenda of our lecture is protein docking using the ClusPro server. The link to the ClusPro server will be provided in the transcription. It is an open access server. First, we will talk a little bit about the ClusPro server. So here you can see the ClusPro represents the first. It is the first fully automated web-based program for computational docking of protein structures. Here the users may upload the coordinate files of two proteins through ClusPro web interface or enter the PDB codes for the respective structure, which ClusPro will then download from the PDB server. How does it happen that how do you upload a protein structure and how do you provide a PDB code? We will talk about that in a moment. The docking algorithms evaluate billions of putative complexes retaining a preset number with favorable surface complementarities. It creates billions of complexes, but out of these only those complexes with favorable complementarities are selected and then filtered. A filtering method is then applied to this set of structures, and only those structures are selected which have good electrostatic and desolvation free energy for further clustering, and the program output is a short list of putative complexes. These complexes, docking complexes, are then ranked according to their clustering properties, which is automatically sent back to the user via email. If you need further information on the ClusPro server and how it works in detail, then you can refer to this paper, and it will also be provided in the transcription. We will close that for now and move on to the lecture. So here we click on, once you click on this link, you will get to this page. Here you have two options, login and sign up. In the login option, you can see here I have already created an ID, so I do not need to create another ID. Simply I will click on login, but if you do not have an ID, then you have to click on sign up. Once you click on sign up, you will see this detail requirement for the account. You have to put your name, your last name, your university, and the username that you will need, and your email ID. Make sure this is an educational ID. Any other ID would not get you the account created. Then you have to type the following number or the word given to you, and then click on I agree to the use of ClusPro only for non-commercial purposes, and then create account. You will get an email in your inbox once you have clicked on the link, the verification link. You will get your password and your for your username. So then you can get here to the login page and log in to the server. So we will close that right now, and once you click on login, you will get to this page. Here in this page you can see you have multiple options. We will close the advanced option for now. So here you can see, here you have to put your job name. For example, I am doing the TLR2 docking with the vaccine protein. For this lecture, I have selected the toll-like receptor 2 as our receptor protein and the vaccine as our ligand protein. So I have given a name TLR2 vaccine. You can click either on CPU or on GPU. Here you have to upload the receptor and the ligand PDB ID. For the ClusPro do is it will automatically download the receptor and the ligand from the PDB database, and then will continue with the docking process. You can upload it from your system. So here in this case I will be uploading that from my system, so I will click on upload, choose the file. Here our receptor protein is receptor TLR2, so click on open, and now click on the ligand protein vaccine. Okay, now you have to specify the chains here. If there are more than one chain, you have to specify that. For example, if there are chain A and then B and then C chain, then you have to specify, and your ligands have only one chain, then you have to specify it as A. But in this case, our receptor and our ligand both have one chain, so I do not need to specify the chains. and then click on doc. Once you click on doc, you can see here the TLR2 vaccine, the ID given to your query, the name of the docking that you gave, your username, and the status. Once it is completed, you will see it here. You can get the results via email, or you can click on here on the queue, and you will be provided with the status of your docking. So here you can see now it says copying to supercomputer and then running and then complete. When it is complete, then you can click on the ID and get the results easily. Before you continue with the docking using the ClusPro, make sure that your receptor and ligand proteins both are ready for docking. If that's not ready for docking, then you have to do some modification and changes. So I will be using the molecular operating environment software for correction and some editing of the ligand and the receptor protein. So I will open that here, the 5D3I that I have downloaded from the protein data bank. I will open it here. Click on the biomolecule assembly as usual in the symmetry drop down list and click on OK. You can see there is an extra chain here attached to the ligand protein, and other than that a ligand is also present in some water molecule, so I have to remove that first. So here you can see 4 ligands are attached, so I will click on delete and delete 5 ligands, and then I will click on this chain, this is the extra chain, and click on delete. Once it is deleted, then I have to do the quick preparation. What the quick preparation do is, it will fix the protonation, it will fix the bond angles, bond lengths, and other inaccuracies with the structure. And then you have to save the file. You have to save the file here. Once this is completed, then you have to save the file, for example, ready file, or you can say final receptor. Okay, make sure you click on the PDB because ClusPro only accepts the files in the PDB format. So then click on OK and your file is ready for docking. So this is how you prepare the receptor and ligand, and same way you have to prepare your ligand protein as well, and then go on for the docking as I described. Okay, so the next thing is that you have to, once your both the receptor and ligand is prepared, you have to upload it as I did a moment ago. Okay, the next thing is that we have some advanced options here. Here in the advanced option, the first option is attraction and repulsion. If you want to know about further details, here you can click on help and then go on to here. Here you will see what the constants, the attraction and repulsion mean, what the tails and unstructured residues mean, what the antibody means, and other advanced options. So I will tell you a little bit about this. So here you can see what attraction and repulsion means. The attraction means that the residues that you think that will participate in the docking. That means the active site. Actually you are providing the active site here. The active site format is this one. You have to provide the active site in the chain. For example, here I will provide the, for example, our receptor, that is chain B. Yes, that is chain A, sorry. So I will click on A and then 35 and then space and then A and then 36 and then space A 37, and then make sure you put the dash in between, and so on. These are the ligand and active sites, and the receptor and active sites. And here in this box you have to provide the ligand active site. For example, that is chain B, so I will put 26 and then B 28 and then B 36, etc., and so on. This is how I will provide the active sites here and to the ClusPro, where the docking will be restricted to these residues. For now I do not have any specific residues, so I will clear this. The next thing that you can do here is repulsion. What here the repulsion means is those residues that you think are not should not participate in the docking. So in here you have to select all those residues that you think should be excluded from the docking. So what the server will do is it will just remove those residues from the docking. The next thing is here you can use the PDB masking file. So let me tell you what the PDB masking file means. So let's get to the ClusPro and click on the ligand protein. For that you will use the molecular operating environment, the PyMol software. So here you can see this is the ligand protein, and we are masking some of the residues. Masking means that these residues should be ignored while docking. So here at the bottom you see S, click on S, and here you see the sequence of the ligand protein. So here you have to select those residues that you think should not participate in the docking. So these residues will then be masked. Here you can see a selection is made. Click on here and export molecule. In export molecule you have to click on enable and then select. Okay, so leave that for now and then click on save. Once you click on save you will get the molecule ready in a PDB format. We will save it here. We will give a name, masking residues, and then click on now. Close that. If you have any residue that should be masked in the receptor protein, you can do the same and save the file. Here you can see these are the masking residues. Now that I want the ClusPro server to avoid. So these were in the ligand, so I will, in the ligand portion, I will choose these masking residues and upload it. And the same you can do for the receptor protein, mask the residues that you think are not participating. The same way in PyMod, save it and upload it here. So what this option do is that the ClusPro server will restrict the docking to the residues that you provide in the attraction. On this side you can see this is for the receptor and this is for the ligand. And same way, if you think that some of the residues should be ignored while docking, so you can either type the residues in these boxes, or you can do the masking such as I did in the PyMol, and then upload those PDB file that you saved. So this is about the attraction and repulsion advanced option, and then the structure modification. Here what the structure modification do is that sometime when we purify the proteins in real time, and so we attach small tag sequences so that we can easily purify the protein. So here what this option do is that when you click on from receptor and click on from ligand, so it will detach, it will remove, it will chop up all those tag sequences. Or if there are some unstructured sequences that are not well structured, such as in the N termini and the C termini, most of the residues do not get structured well. So this option will remove and chop up those atoms that unstructured terminal residues. So and the next option here is a structure modification. The multimer docking. What the multimer docking do here? Okay, so here you can see the multimer docking. What the multimer docking do here is that when you have two proteins and that are exactly similar, and you want to dock those two proteins, then you have to click on this. Then you have to click on this multimer docking, and here you have to specify the number of subunits and this protein that you have uploaded. So here only one file will be uploaded, and that will be in the receptor option. In here you can see it supports dimers and trimers till now. So here you have to set if there are 2 subunits or if there are 3 subunits. So you can click on that and it will automatically generate a copy of the file that you have uploaded, and then those two files will be docked with each other. The next option is antibody mode. Use the antibody mode. So when you click on the antibody mode, here you have to provide the receptor mask of the non-CDR region. What actually happens in the antibody and the antigen docking is that these are docked based on complementarity determining regions. Here you can see in antibodies binding usually occurs to the complementarity determining region. So you have to provide the details about which residues are CDR and which are non-CDRs in case you are doing an antibody antigen docking. So this was about the antibody antigen docking. Okay, and for the rest of these are not important right now for us, and we will discuss that in our peptide docking tutorial using the ClusPro server. So we will leave that for that lecture. So let's know about whether our structure is completed or not. So click on Wave, and here you can see the structure is running on supercomputer. So we will wait for that and come back when our structure or docking is complete. And now we are back with our results or docking results. When you open the ClusPro, you see this page, and these are the jobs that are still running on the computer, and different people have submitted different jobs. We will go to the results, and once you click on this, you will get to this page. In this page, you can see here the ID that was assigned to my docking job that I submitted, and here you can see the TLR2 vaccine that I named it. The vaccine is the ligand protein and the TLR2 is our receptor protein, and you can see here our job is finished. These are some of the previous, you can see these are some of the previous jobs that I have done using the ClusPro server, and it was really good and appreciated. Okay, so now when you click on this ID, you will get 10 models displayed here by default, and we are provided with 30 models. So I will click on 30, and so all of these 30 models will be visible here. Here you can see these are the 30 different models. These are just the pictures. Here you can see you can click on view model scores. You can click on download R per coefficients. You can look for the electrostatic favored, hydrophobic, van der Waals plus electrostatic, etc. And when you click on the download displayed models, you will get all of the models downloaded. So here you can see I have already downloaded, so I do not need to download it again. So we will close that. So now we will click on the view model scores. So in view model scores, you see this table, a center here minus 1359.5, and the lowest score minus 1359.5, in each of the cluster that have center and lowest score, Here we prefer those complexes that have the highest number